A fundamental process in all of life is cell growth. For decades, a central area of research has been to define the mathematical nature of this growth. Does a cell accumulate mass at a constant linear rate, or does its growth accelerate as it gets larger, following an exponential pattern? Fundamentally, cell growth is the creation of new biomass, the polymerization of small molecules into the lipids, proteins, and RNA that make up the membrane, cytoplasm, and organelles. The primary obstacle has been measurement of mass. Most early methods like microscopy measure a cell's volume, but volume can be a deceptive metric as it can change rapidly due to osmotic shifts without reflecting a true increase in biomass. Until recently, existing technology lacked the sensitivity to continuously track the mass of a single living cell, which is on the order of picograms and requires femtogram level precision to monitor. This challenge was overcome by the Suspended Microchannel Resonator, or SMR for short. This system consists of a microscopic hollow cantilever that is vibrated at its natural resonant frequency. As a single cell suspended in fluid is pumped through the embedded channel, its mass is momentarily added to the cantilever. This added mass lowers the resonant frequency of the system. The device measures the cell's buoyant mass, which is the cell's true mass minus the mass of the fluid it displaces, and is an excellent proxy for the dry mass of the cell. The SMR is sensitive enough to detect the minute frequency shift caused by a single cell, allowing for mass measurement with femtogram resolution. To measure growth, a single data point is insufficient. For this, the authors engineered a dynamic trap. A feedback control system detects the cell as it exits the cantilever and immediately reverses the direction of fluid flow. This reintroduces the same cell into the resonator for consecutive measurements. This technique allows for the continuous monitoring of a single cell's mass over extended periods, all while the cell remains in a nutrient-rich medium, permitting normal physiological growth. By applying a linear fit to the buoyant mass data of trapped cells on a graph of mass versus time, they could determine the instantaneous growth rate from the slope of the best fit line. Across diverse cell types, a linear relation was found between a cell's initial mass and its growth rate. We can see here that heavier cells grew faster. The relationship between cell size and growth rate appeared to be linear, and for B subtilis, the linear fit extrapolated close to the origin, which is suggestive of a simple exponential growth pattern. To ensure the results weren't skewed by accidentally measuring clumps of cells instead of single ones, the authors created a computer simulation that mirrored their experiment. A fit for exponential growth, even with virtual cell clumps, matched their real data for B subtilis and gave them high confidence in their observed exponential trend. Furthermore, the high precision of the SMR uncovered significant biological variability. Cells of the same mass did not always grow at the same rate, which suggests growth could be modulated by the cell's position in the division cycle or stochastic fluctuations in gene expression. This approach can contribute to the study of many cellular processes, and according to the authors, future versions of this system will provide even more experimental power.